Diversity Consultant with the IFC. And I welcome you all to the 5th webinar in this IFC webinar series for Nepal on advancing sustainable hydropower with a focus on biodiversity assessment and management. So we're very happy to have you with us, whether you're in the evening, afternoon, morning. Namaste, and happy that you're joining us today about environmental DNA. And we're very pleased today to have two speakers who have entrepreneurs in this field and leading specialists in environmental DNA. So we are very thankful that they're joined us today to share their knowledge with us. So this series is aimed to bring together all kinds of different diverse stakeholders in order to share with each other, learn, and discuss together how we can achieve sustainable hydropower. But primarily with a focus on how do we really understand biodiversity, get a good baseline, and how do we monitor it over time to assess impacts. This series is part of IFC's Hydro Advisory Program for Nepal that's led by Kate Lazarus. And it's generously supported by the governments of Australia, Japan, and Norway. And so we thank them for their support of all the work that IFC's Hydro Advisory is able to do in Nepal. So before we start today, there's me, just so you know who I am. I'm here again, so you probably have seen me. Just remind you that this webinar is part of a 6-webinar workshop on the components of the Trishuli assessment tool, which is a standardized field methodology for aquatic biodiversity assessment and monitoring that a group of researchers in Nepal and international scientists have been working on in order to improve biodiversity assessment and monitoring for hydropower projects. So we had first an overview of the tool, and last week we heard about electrofishing from Bill Beaumont, and today we're on environmental DNA. On Thursday we'll hear about macroinvertebrate sampling, and then next week we will finish up this 6-webinar workshop with more about fish taxonomy and identification, and then about data analysis for the long-term monitoring, which is a key component and if we're going to actually be able to see and determine the changes. And as before, if you go to all 6 webinars, you will receive a certificate from the IFC and be considered for the in-person training that we hope to have on all of these topics as soon as things are free up so we can do more field work. So I just want to announce, though, we have one additional webinar in our series, which will be on Tuesday, March 23. So we had a lot of interest in a previous webinar about the hatcheries. So David Phillip and Julie Clausen, who will be leading the webinar in telemetry on March 16th, they have generously offered to give another webinar about hatcheries, a review of the costs and benefits of hatcheries and how to do it in the best way. So thank you, David and Julie, and that will be on March 23, and we'll send out a flyer. So, excuse me, so you'll have that information and the link. So just for today's webinar, it's an hour and a half, including the question and answer. You'll be automatically muted during the webinar, and we will be recording it, so please be aware of that. Please don't use the raise hand option. If you have any questions, put them in the chat. And I will be collating the questions and answering and posing the questions to both speakers after the 2 speakers have delivered their talks. If you need any help during the presentation, having any trouble, please do a private chat to Upasana Pradhan, and she will help you out. And I'd like to thank Upasana so much for helping to put together this whole webinar series and for taking care of us during the webinar, making sure it runs very smoothly. We will share the PowerPoint and the recording of this webinar and all the webinars with the participants afterwards. So you'll get that. And we'd appreciate if you would fill out the evaluation of the webinar that you will receive by email afterwards. So the agenda for today, after I am finished soon, we will first hear from Kat Bruce from Nature Metrics. Then we will have a talk from Debesh Karmacharya from CMDN in Nepal. And then we will do the Q&A, as I mentioned, all together afterwards. And if we have time, we'll open the floor so people can ask their questions directly. Let's see. So without further ado, I'm going to turn it over to Dr. Kat Bruce to give her presentation. And if she wants to load her slides while I introduce her. So Kat trained as a tropical ecologist, making many trips to the Amazon rainforest in Ecuador and Peru. She obtained a bachelor's degree in wildlife ecology from Anglia Ruskin University, a master's in entomology from Imperial College, and a PhD in molecular biodiversity assessment from the University of East Anglia, which I assume all these are in the UK. Man, I want to get a PhD in molecular biodiversity assessment. That sounds awesome. After completing her PhD in 2014, she set up Nature Metrics to bridge the gap between the powerful molecular tools being developed in the research world and the environmental managers who could benefit from their use. Her particular interest lies in how to bring together the worlds of research, industry, and policy to drive forward advances in our capacity to monitor the natural world. Through her work with the EU Cost Action Project, DNA Aquanet, I'm not sure if that's right, she works to establish best practices for the use of DNA monitoring tools for aquatic biomonitoring. So we really have a very great specialist in this topic to share with us today. So thank you, Kat, and welcome.
Thanks very much. It's really a pleasure to be invited to speak today. So thanks, Leanne. As Leanne mentioned, I'm the founder of Nature Metrics, and we're a UK-based company that specialises in delivering eDNA services for projects all over the world. I've also been very heavily involved in the research community in Europe and in also starting to set the standards for this new industry. It's a really exciting time to be using these tools and doing what we're doing. So today, today I'm going to introduce a bit of the background to environmental DNA and metabarcoding, which is the sort of molecular method that we use for analysing it. And I'll introduce Nature Metrics and our systems that we've set up for the projects that we work on, and then show you some data from some case studies from around the world, which sort of help us to build confidence in these tools and techniques and the data that they give us. So the core tool that we use at Nature Metrics is called DNA metabarcoding. And this is basically a way of generating a list of species from complex samples that contain many different species in a single reaction. So you take your sample, you extract all of the community DNA from it, so you have a sort of soup of DNA, and you sequence that DNA on a high throughput DNA sequencing platform. That spits out millions of DNA sequences, which are then processed and matched against reference libraries of known species to identify them. And we can apply this basic method to lots of different types of complex samples. But the three samples that we work most with are collections of insects, soils and sediment samples, and water. And from each of these different types of samples, we're able to look for many different taxonomic groups. So you can start to build up really comprehensive biodiversity data sets from a very, very efficient sampling regime. And that's one of the major strengths of this technology. But for today, we're going to focus on water samples and environmental DNA. Environmental DNA, or eDNA as a term, refers to any time when we're not actually catching the organisms themselves, we're working with traces that the organisms have left behind in the environment. Mostly when we talk about eDNA, we are talking about DNA from water, but animals do leave eDNA traces in other types of samples as well. But today we're just sticking to water. So we know that when animals are in or around water, they're shedding cells that contain DNA. So those are through mucus, skin, feces, urine, all sorts of bodily processes and excrements. And so the water becomes like a sort of soup of the DNA of the wildlife that's been in and around it. And that eDNA remains viable for a few days for identifying the species that it came from. And we can capture it from the water just by passing the water through a filter, and the DNA gets stuck on the filter membrane. That filter can then be sent to the laboratory, and the DNA is extracted from it. So you end up with sort of a small amount of colorless liquid in a tube, and that contains the DNA of the whole biome from that water sample. So it's got everything in there from the microbes up to the mammals. We then have to amplify the DNA. So at this point, we have to choose what taxonomic group we're going to focus on, and we make millions of copies of the DNA that's come from that group so that we've got enough to sequence. And you can choose to target very broad groups or very narrow ones. And what you choose depends very much on the study that you're doing. So we spend a lot of time working with our clients to help them choose the right assay or set of assays for their project. But you can see here the three assays that we use very frequently. Either you can target sort of fish and mammals independently, and we use different genes for targeting each of those. So 12S gene for fish, or part of the 16S gene for mammals. Or we can use a broader sort of whole vertebrate assay using part of the 12S gene. And that wider vertebrate assay will give you a sort of broader cross-section of the total vertebrate diversity in the samples, but it's more likely to miss some of the rarer species from both groups. So there are trade-offs in making these decisions. Now, the good news is that when you run that first analysis, you actually only use up a little bit of the DNA that you've extracted from the filter. So there's almost always plenty left over, and we can run additional analyses on the same sample. So once you've amplified your DNA for your chosen target group, then we sequence it on a high throughput DNA sequencer, like this Illumina MiSeq, which is just a sort of benchtop machine. And that takes about 2.5 days to run normally. And we normally put about 100 samples on at a time. The sequencer spits out about 30 million DNA sequences every time it runs. So we've got a couple of 100,000 sequences for each of the samples that we've put on it. Now, the sequences that you get back, most of those are exact copies of each other. So you might have 100,000 copies of the exact same sequence. So the data goes through a processing pipeline called a bioinformatics pipeline. And what that does in the first instance is it applies a quality filter. So it gets rid of any poor quality sequences. And the ones that are left over, it summarizes into this table. So it tells you for each unique sequence, how many copies of it you've got in each of the samples that were on your sequencing run. And then it takes each of those sequences and it compares the sequence against a reference library of known species to identify it. So we end up with something that basically looks like a species by sample table. And the numbers in each of your cells tell you how many copies of the sequence you have for that species in that sample. And what we find is that, especially for species that are giving off eDNA in quite a consistent way, so things like fish that are in the water continually shedding eDNA, these numbers give us a pretty good representation of the relative abundance of the different species. So Naturemetrics is the largest specialist eDNA company in the world. We've been running since 2016. We have staff who cover everything from the molecular laboratory through to ecologists and conservationists and biodiversity scientists who work with our clients to design and deliver the projects. And we work all over the world, even though we're based in the UK, working with quite a wide range of different clients, from sort of large energy mining infrastructure clients through to the conservation organisations at sort of national and international levels. And we provide an end-to-end service. So that involves everything from providing sampling kits, which I'll talk a bit more about in a second, through the full sort of laboratory workflows. We've got an in-house team of bioinformaticians and reporting. And this is all held together by an online ordering and tracking system where you can place your order and then track the progress of your samples as they progress through the system and access your data in there as well. Our sampling is based on our providing kits that our clients use in the field. So we don't send teams out to do the sampling ourselves, although we do quite often for the larger projects accompany the field team in the first instance to provide sort of detailed guidance and training in that first season. But generally speaking, we've got kits that can be used by our clients. And we've designed these kits with a really simple vision in mind, and that is that this is an amazing opportunity to really democratize the acquisition of biodiversity data. So we wanted to design something that could be used by anybody anywhere in the world to collect high quality eDNA samples. So we've custom designed an eDNA filter, and the filter membrane itself is contained within this plastic disc housing. And that makes it very difficult to contaminate. So that's one of the big risks with eDNA, because we're working with such tiny traces of DNA that they're very vulnerable to being overwhelmed by external sources of DNA. So that's very difficult to do with these enclosed filters. It makes it really robust against contamination. And basically it works just by pulling water up in a large syringe. The syringe screws onto the filter and you push the water through. So the water comes out the other side, either onto the ground or back into the river or lake it came from. And the particulate matter and DNA is trapped inside this housing. And you push through as much water as you can. Because of the way we've designed the filter, you can push quite a lot more water through this one than you can through the filters that you can buy off the shelf, which is why we designed this. You then need to stabilize the DNA so that it doesn't degrade on the way to the lab. So that's done with a preservative solution in the kits, which is in a small syringe. It just presses inside the filter housing and you pop a cap on each end. And the preservative is designed to work at ambient temperature, even in the tropics. So we've tested this for up to about 3 months. So it means you don't need fridges and freezers and things to keep the DNA safe. And then the samples are sent back to the lab, and we've got an in-house team who can help with that logistics and export of samples where needed. So we recently tested out this assumption that anybody could use this process to collect good quality eDNA samples using a group of citizen scientists in the UK. So we all went down to a river with this group. None of them had ever taken an eDNA sample before. So I sort of quickly showed them how to do it. And then each of them took one sample from the river, and I took 2 samples at the same time for comparison, because after all this time, I should know how to do it pretty well and get good quality data. So this is the results. So each of the columns is one sample taken by one of our participants, and my 2 samples are over on the right-hand side. And each row is a species, and the size of the bubble is proportionate to the number of sequences. So I think, the results speak for themselves. There's no discernible difference between the data that we got from the samples that I took and the data that the participants got. Pretty much every single species is detected in every sample, and in actually really consistent relative abundances. So that actually gives us just a huge amount of confidence in the quality of the data that we're getting from these samples, even when the samples are collected by people who haven't done a lot of it before. And there are huge advantages to this. So it means that, for example, projects can save a lot of money by hiring local contractors to do the sampling, train them up, and you've got people who can collect good quality samples. That means we can sample much more regularly, because you're not having to only collect data when you've sort of deployed a full team of ecologists. You can use people who are on site on a permanent or regular basis to be collecting eDNA samples year round and actually build much better insights as to how communities are changing over the course of a year, and that can inform your management and mitigation. It's a great opportunity to engage local stakeholders because you can get local communities involved, you can get local schools involved. We've done this with children aged 5, 6, 7. Everybody really enjoys doing it, and you can tell some great stories about the wildlife and the species that are found. And it also gives opportunities for auditing, because you've now got something which is a very standardized tool that different teams of people can independently use to get the same data. So, how does eDNA compare to conventional survey methods? This was one of the first questions that we really set out to address when we started up the company, and spent a lot of time doing side-by-side studies in Europe in the first couple of years. In particular, we worked very closely with a company in Sweden called Aquabiota, who had just huge amounts of information and knowledge about fish communities in a set of Swedish ecosystems. And broadly speaking, every time we've done a head-to-head, the eDNA comes out as being far, far more sensitive than any conventional method. So whether that's netting or electrofishing or test fishing or historical records. And so, this was an example in a Swedish hydropower project. They took samples at 3 different points, and 2 days of eDNA effort detected far more diversity than sort of having aggregated all of the information that they had from the last 30 years of electrofishing and netting data. And all of those records were then subsequently verified. So these are real detections. And we actually find that, when we look at eDNA versus electrofishing in rivers in the UK, where our Environment Agency does regular electrofishing every year or 2 years, we find that 1 litre of river water provides as much data on fish as about 10 years of cumulative electrofishing data from the Environment Agency. And we see similar sort of patterns in the sea as well. So this was, again, working with Aquabiota in Sweden, head-to-head with overnight netting and 1 eDNA sample in each point. And again, in each case, we're finding more of the total diversity in the eDNA sample than we caught in the nets. And actually that becomes particularly true as the water gets deeper and the efficiency of eDNA over the netting is more pronounced. So in the UK and Europe, we're now in a situation where actually eDNA metabarcoding is very, very well validated for fish surveys. It's starting to be taken up for regular routine monitoring by governments here. And it's supported by a lot of published literature, and we can identify all of the species with high confidence because we don't have very many species, and we know a lot about them. They're all in the reference databases. Of course, things are very different when we go into tropical environments. And I think that's really sort of highlighted by this tiny little project that we did with a local stakeholder training session in Liberia. Where each of so we had a group of people who got together for a workshop who included sort of local conservationists and members of the Environment Ministry. And we went out and sort of showed them how the eDNA worked, and everyone had a go at taking samples. They divided into 3 groups, and each group took 1 sample just from the sort of urban waterways in Monrovia. I think you see the data on the right-hand side there. You see very similar patterns overall to what we saw with the citizen scientists in the UK, where we've got very replicable data. All the species pretty much are showing up in all of the samples and very replicable relative abundances. But the big difference is that we can only name a small portion of the species down to species level. And that's because the reference libraries here are really, really incomplete. But again, we've sort of demonstrated that non-experts can collect really good quality replicable samples. And this issue about the incomplete reference databases is actually less of an obstacle than people often think. So this is a data set from actually a similar area in West Africa as part of an EIA project. And you've got your samples over on the right-hand side with the numbers of sequences and the taxonomic information on the left. And you can see here that actually when we first ran this analysis and just compared everything against the globally sort of available reference libraries, we couldn't identify anything here to species level with high enough confidence. But in this instance, because it was a pilot project and they were doing some netting at the same time for comparison, when they caught and identified fish in the nets, they just used a very simple swabbing kit to collect DNA from those samples. Those swabs were sequenced, so we generated new local DNA references. When we added those to the library, all of a sudden we were already able to identify a big proportion of the species that we're finding in the eDNA. And the nice thing about the eDNA is that, even if we can't identify things to species now, we've still recorded it and we have that sequence. And that means that as the reference libraries grow, we can just keep going back to the data we've already got, rerunning the taxonomic identification and adding on the new names. So the amount of information in these data sets grows over time with no extra effort. And we actually just heard that there was a big project just funded last week to do a big effort to barcode West African fish. So we're really excited about that. I wanted to show you a larger scale project here to sort of give you an idea of the power of this tool for use at landscape level for doing biodiversity surveys. This is a project that we did with WWF in the northern Peruvian Amazon. And they really wanted to know whether eDNA could help them monitor two key species, so pink river dolphins and Amazon manatees. So we thought, well, rather than just looking for those species and being very narrow, why don't we do a broader assay and look for vertebrates in general? That should give you those species, and we can run a check beforehand to check that we've got good reference sequences and that we should identify them in the vertebrate assay. So they took four half-litre water samples at each of 40 points across this area. It's about 500 kilometres from west to east here. We sequenced them for vertebrates using the 12S gene. So the first thing we looked at was, we were able to find these species and we can map their distribution across the landscape here. So the numbers in the circles show you how many of those 4 replicate samples the species was detected in at each point. So you can see with dolphins, we get loads of DNA. There's lots of dolphins. We saw them everywhere, so it wasn't a surprise. And so we're picking them up in every sample, basically. With the manatees, it's a really different picture. There are many fewer of them, they're less active. And so we're only picking up tiny bits of DNA from them. And you sometimes only get them in 1 or 2 out of the replicate samples. So you can start to get an idea of how rare or common things are based on this sort of frequency or occupancy at the landscape level. But in addition to these species, we found over 675 vertebrate species in general. So this was for absolutely no extra effort. And doing their conventional surveys, they would have got data on just the dolphins, and we've got data on 675 species. And what was really surprising in this data set, so this was a few years ago, it's the first time we'd really done a study at this sort of scale. And what surprised us was the amount of data that we got on terrestrial species. So it's not just the things that are in the water, but we've actually been able to then map the detections of over 150 species of terrestrial mammals in this area. And that includes, all of the monkeys known from the area. It includes, I think, about 25 species of bats. We've got really rare things like jaguars showing up. And so, the amount of effort that it would have taken to generate this level of data on so many species at this sort of landscape level is just unthinkable, really. It wouldn't have been done. And in terms, most of the data that we got were fish. Again, like in West Africa, most of them can't be identified to species level, so only about 20% of them could be. But even then, we were actually able to get a huge amount of ecological information, even in the absence of the species names. So in this case, we were able to really show how the communities of fish are changing across the landscape and sort of varying in different parts of it, and also in different habitats within that landscape. And we can also detect where there are very abrupt sort of sudden changes in species communities, which are linked to things like barriers. So in this case, we've been able to show there's a big impact of a natural barrier, which is a sort of gorge and a set of rapids. And you have very distinct communities upstream and downstream of that. But it would be exactly the same process with dams or man-made barriers. So when we've got lots and lots of data, basically, we can start to map ecosystems across the landscape. And this project was approximately a sort of $50,000 project to obtain data on 675 vertebrate species at this landscape level in Peru. We've also done some in this sort of same area. We've been able to do some head-to-heads to compare the eDNA against the conventional fishing methods. So this was part of an EIA project with Repsol in the Peruvian Amazon. We took eDNA samples alongside the netting that they did for fishing. And overall, I mean, it was quite a small scale project, so only about 24 samples, I think. But we detected sort of 5 times as many fish species in the eDNA compared with the netting. And what was interesting here is that that's not the same across all of the different environments. So in small streams, the netting is quite efficient, and actually you get quite similar levels of diversity from the two methods. It's in the rivers that the eDNA has a huge advantage, because the netting is just really, really inefficient in the rivers, only picking up a tiny proportion of what's there. And we've seen that time and again across different high diversity systems. And then, so this was actually from the same data set. So we did this head-to-head to look at the fish, but we thought, well, since we've got the samples already, let's look at the other vertebrates as well. And just in that sort of bycatch almost from the fish data, we've been able to get as much as or more information on the other vertebrate groups as was obtained from the sort of targeted conventional surveys. It's not necessarily the same species, because the things that show up in the river may be different from what you find on land, but it's a huge amount of information, and we can get this information even in the wet season when they haven't been able to mobilize teams to go out and collect and do the conventional surveys. And overall, there were 12 IUCN red-listed species that we detected in the eDNA data that weren't detected by the conventional methods at all. So that's really valuable additional information. And this was part of a much sort of broader pilot study, which included soil sampling and insect trapping and all sorts of different things. But this particular component of it using the eDNA was approximately sort of $12,000 project. So hopefully that's sort of given you a sense that the eDNA is a really powerful tool. It is increasingly being sort of well validated. There's a huge scientific community working on this, and we're part of that. And it is something that is really transforming our potential to monitor biodiversity, particularly because you can put it in the hands of different users all over the world. So thanks very much for listening, and I will pass over.
Great, thank you very much, Kat. Very interesting, and such a broad range of examples and breadth of experience. So I'm sure, thank you very much for sharing with us, and I'm sure that we're going to have a good discussion. There's already many questions in the chat that I will try to compile so for the Q&A session. We'll try to get to all of them. So next, though, I'd like to turn this over now to Debesh. So Debesh, if you want to load your slides while I introduce you, please. Great, then I want to share my screen now. So Debesh Karmacharya, he's the Executive Director and Principal Investigator at the Center for Molecular Dynamics Nepal, or CMDN as we affectionately call it. He has a conservation biology degree from Wayne State College in the USA, and a PhD in conservation and microbiome genetics from Griffith University, Australia. He worked in the US for Caliper Life Sciences and GE Healthcare Life Sciences. And then he founded the Center for Molecular Dynamics Nepal, which is a wildlife genetics and clinical epidemiology research center, of which he's the chairman and executive director. He also founded Intrepid Nepal Private Limited, a molecular diagnostics-based biotechnology company, and Intrepid Cancer Diagnostics, a leading cancer diagnostic laboratory. He's the founder director of BioVac Nepal, a vaccine research, development, and manufacturing company. He's helped to build Nepal's first genetic database of wild tigers through the Nepal Tiger Genome Project, and is developing eDNA technology for aquatic biodiversity assessment. He's also the principal investigator of Predict Nepal project, It's an emerging pandemic threat project, and he leads several genomics projects in SARS-2 and COVID-19. So we're very happy to have Debesh with us, because I'm sure he's very busy these days working on vaccines, I hope, and COVID research and all kinds of very interesting and important research. And we're so happy to have him with us today to share with us. So welcome, Debesh.
Thank you so much, Julianne, for inviting me to present this work. And first of all, I think I would like to, before I even start, thank all the scientists who came over to Nepal and really worked with us to develop this technology in Nepal. So what I'm going to present today is actually a project that we did with IFC, with other partners and collaborators. We basically use this study to get information about fish biodiversity in Trishuli River. And then I will also kind of present a few data in terms of the story of two rivers, Karnali and Trishuli, which is quite interesting. When we were actually working or putting together eDNA capability in Nepal, we started back in 2014 with a project called Nepal Fish Biodiversity Project. It was a project funded by USAID and worked with US Forest Services. That was done to do a couple of things. One, it was really a capacity building to build a genomic level facility to carry out eDNA in the country. And secondly, it was really a pilot project to see the feasibility of eDNA in aquatic biodiversity assessment in particular, but biodiversity in general. And we were able to build a genetic database of fish, Nepali fish species in Karnali that really helped us to kind of move further with the technology to be used in environmental impact assessment kind of effort, which is the case with the Trishuli project that we did. So Trishuli River is one of the major rivers in Nepal. It is also a river that has been heavily, from a hydropower perspective, heavily developed river. And when IFC was considering a project of a hydropower, we needed to kind of do an environmental impact of the site and of the river. And this is how the project kind of started. And initially, there was a lot of consultation with the IFC experts, as well as our experts from within Nepal, in terms of figuring out how to kind of do this, right? And so we wanted to kind of look at a hydropower, proposed hydropower site, and the upstream of it and downstream of it, and try to use eDNA to figure out how many different types of species, fish species there are. Obviously, when we were trying to do this, one of the eDNA is as good as of a technology as long as it has a really good reference database. There are larger, broader global reference database like NCBI, but a lot of the fish species are not reported from Nepal and dumped into that database. So at that time, we felt like, not only we needed to use eDNA to see if we can kind of detect fish species, but we really needed to validate that against catching the fish and actually looking for it and kind of doing a comparative study as well. So, so we actually chose in this first phase, and there are two phases of the project that we did. In the first phase, we actually went out and kind of looked at 7 different sites within the river system. Upstream, some of them were upstream, and some of them were downstream. And then while we were at it, we also wanted to kind of get a broader ecological picture of the river system and the ecology it supported. And field work is always the hardest, and it takes a lot of effort to do the field work. But while we're at it, we can also gather other kinds of information as well. And so not only we were able to look for fish diversity, but we also wanted to kind of look into more of a habitat, water quality, macroinvertebrates. And I believe there is another session that talks about macroinvertebrates, and other information as well. So we wanted to kind of have a holistic approach in terms of gathering information as much as we could. And this is done for the first time. And therefore, there was these were also a baseline information in many respects on this. And so we worked with our other collaborative partners like Kathmandu University, Tribhuvan University, and other experts within Nepal and outside to kind of put together all this different information. And like I mentioned before, the reference database coming from Nepal, there are still a lot of different gaps, and therefore it is important that we kind of go out and look for these species and try to get hold of it and get the voucher specimen for it. And so we actually, on this particular study, we actually went out and caught fish. We caught about 125 different fish using cast nets, angling, gill nets, that sort of thing. And we brought about 65 of those as a voucher specimen to our lab. And then we identified the species using DNA barcoding on these. And this became part of our reference database. Again, like I said, it was a concerted effort of not only just collecting water and for the eDNA purposes, but also looking at other different things, including voucher specimen, including building that water quality, water parameters stuff, and of course eDNA as well. And our system of collecting water and eDNA is a little bit more cumbersome, it looks like, as opposed to what Kate has put together with the syringe and little disc. Ours is more like we have a pumping system, handheld pump, and we kind of get 2 liters of water, go through a filter, and then that filter will then capture all the tissues and biological stuff that's been shed in the river, and then from that we then extract DNA, which I'll talk a little bit about. And then on the voucher specimen itself, we bring it to our lab, and we actually work with, so we're not fish experts, right? And so we work with a lot of fish taxonomists and fish experts in order to basically identify the species. And we meticulously kind of take representative voucher specimens and keep it in our what do you call reference voucher cabinet. So over the years, between 3 different efforts, we have more than 70 different species of fish collected from 2 different river systems. And of course, we've been inputting the fish DNA barcode information into a global database such as NCBI. I'm not going to go through this slide because this basically is what Kate has already covered. Basically, in a river, there are all these slouched up, shedded tissues from all different animals, and then basically, if you collect those cells and tissues and get the DNA, you'd be able to kind of identify what those DNA are coming from, and therefore identify a species. As far as eDNA goes, again, it's not just simply going out to a river and collecting the samples. There are different areas, pockets, these small fish kind of pooled together, and so you basically want to get a broad representative samples from a river that would include pools, that would include riffles, that sort of thing. So when we went out and collected samples, we made sure that all of these different types of small ecosystem within the river are well represented when we were collecting samples. And so our effort was to figure out the aquatic biodiversity using eDNA. Again, taking 2 liters of water, upstream, downstream, and all these different pools and riffles and that sort of things. And so we basically do the filtration out in the field. The picture kind of shows us putting out a table. And then the first thing that we do is we basically pump we'll make sure that there is no cross-contamination. We pump the 2 liters of water through a filter paper, and then that filter paper then gets put in a preservative in a small tube and then sent to the lab. Meanwhile, our taxonomist group and our fish group who catches fish lays out another table on the side and goes through the keys of trying to figure out different species of fish that we caught together. So as far as lab process goes, again, I'm not going to go through this. Basically, we extract the DNA, and then we, whether it's the voucher specimen DNA or tissue DNA or environmental DNA, then we actually do the what do you call a reference voucher specimen species identification using COI marker, CO1 marker. And then on the eDNA, we use actually 12S, as Kate mentioned, and then we do the analysis. So in terms of fish identification, again, voucher specimen, verified species with DNA barcoding, and then we do metabarcoding for environmental DNA assessment. And then we have this elaborate water quality information of different sites. And in terms of eDNA, again, going through a massive amount of data, then pulling them together, going through a bioinformatics pipeline, then we kind of identify different species within. For example, for tuturas, these are the regular fish that we kind of saw in eDNA, and also, they match with the caught species as well. So the other advantage of eDNA is looking at the abundance, right? So the assumption here is if you have more fish, they are going to shed more tissue into the river, and therefore, when you are detecting the eDNA of those, you're going to see a lot more of that species than the others. So technically, you would be able to tell a predominant species having more abundance coming on this eDNA. Like I mentioned before, we have put together a key for species identification out in the field on a morphological level, and we kind of do that, and then the fish that we have identified, then we have done more of a from their ecology perspective and all that, their presence absence perspective from previous studies and all of that. So we kind of tally those numbers and see where they stand. In our effort on the first round of the project, we identified 21 species through eDNA. We identified 9 species through fish capture. And so out of these 2, so in total, there were 6 that were common to both eDNA as well as fish capture. And so, and then one of that species was an endangered species. Again, we have done a lot of these comparative data, site-specific data on the fish detected and that sort of thing. To get a broader picture of the species distribution and the comparison between different sites. On the second round of our effort, we worked with a group of international experts to really compare different tools that are available to do fish assessment, biodiversity assessment. And so, again, we work in the same river system, and we went to many different sites, used different techniques, and we looked into, which technique would work the best to do aquatic biodiversity assessment in general, fish biodiversity assessment in particular, especially from a utility point of view on the EIA kind of work. We, again, looked into 14 different sites on this second round. And again, on this, we only used eDNA on our part, but there were other efforts done by other experts. And we actually detected 16 different taxa from this effort. So all in all, between those two different river systems and all the fish that we have detected, we have created this database and a visual interactive interface so that you could kind of go into a river system and a site and then find out the fish that we have detected, either by capture or by eDNA. And our goal is to build this database of different river systems in Nepal to eventually catalog all the different freshwater species of fish in the country. And this database is basically searchable. We can do, it has all kinds of information, including the DNA sequences as well. So what we have also found, as far as one of the limitations of eDNA is because a lot of the species informations are not in a global database. When we try to do a reference data matching, some of them are not there, and there are a lot of confusion in terms of species identification level and also the eDNA level. So there are some limitations in terms of species level identification, and that's because the resolution of these reference databases are not there yet to kind of get you a very specific species level identification. And like I mentioned before, we now have a comparative database or data sets for two different river systems. One river which is almost pristine with very few hydropower development, and the other one which is very highly developed in terms of hydropowers. So Karnali River has a very few hydropower. It's one of those pristine rivers remaining in the country. And when we did species identification using eDNA, not only we saw variety of different species, more variety of species, but we also saw more abundance of the species as well, compared to Trishuli River, where there are over 30 hydropower development already in place, where we see fewer species and lesser abundance. So this really gives a clear picture in terms of what happens when there are anthropogenic activities in a river system and how that impacts on the aquatic biodiversity. Moving forward, I think where we are going next is really portability. So when we did the eDNA work, either in Karnali or in Trishuli, the field work takes at least 2 to 3 weeks to just go out there, get the whole team out there and do it. Maybe some of the solutions that Kate has proposed might work for us as well. But all in all, the effort is really massive. And so to bring the samples back to the lab and then get the results, you're waiting for at least 3 to 4 weeks before you get the results. Our idea is to make things portable enough so that we can do this out in the field. And this is something that we have done with disease detection. And we think that this might work as well for the fish detection as well. So That's the end of my presentation. A lot of effort, a lot of teamwork, a lot of national and international collaboration. And actually, a lot of this work was done by my team, led by others, and Nikita and our lab team, Jyoti and all. I wasn't that much involved in the field work for this one. But it's really nice to get this collaborative work and help from all over the world so that we could do something a bit like this in Nepal. Really building the baseline information on biodiversity, aquatic biodiversity in this case, and we know that there is still a lot more work to be done to find out more about the species that are out there and how different elements, including climate change, might be impacting the aquatic biodiversity. So with that, I end my presentation. Thank you.
Great, thank you very much, Debesh. Very interesting, and it's great to see a very specific example of how this technique has been used in Nepal and how we can continue to use it. And I'm especially excited about how it's really a collaboration. It's a way to really build collaboration between Nepali researchers, international researchers, but also different disciplines. You need ecologists, you need fish taxonomists, you need the geneticists all together working, and it's been very interesting for us as well. So thank you very much. It's a great study. So there's been many questions. I tried to group them in a couple. I think there's a couple different themes. One is kind of about logistics, about how you actually do the sampling. One is about the reference databases. One's about quantitative sampling and comparisons over time. And another group is on the DNA and how it survives and what it does in the river. And another is about limitations and costs. So I'm going to go through these groups. If you have more questions, put them in the chat, but I hope to get to them. But I'm going to start with the questions we have so far. I think I'll start with the one question that maybe is the hardest one. Is about, Debesh showed the different, what they call the OTUs, right? The actual data you get from the fish, and how you use this. What kind of quantitative data is this? How accurate is it? What kind of abundance data do you actually get with this? And how would you use it to monitor over time? So I don't know, Kat, do you want to start with that in a general sense? And Debesh could talk about how they're using it in Nepal.
Yeah, sure. So I think in a general sense, we started off being really conservative about the quantitative inferences you can make from eDNA. And part of that is because when you do that amplification process of the DNA, you have genetic primers which attach onto either end of the area of DNA you want to amplify, and then they copy it. And those primers are designed to match the target DNA. And they will naturally, if you're targeting quite a broad group of diversity, they will naturally bind on better to some species than others. So that means that some species might amplify more efficiently than others and end up with more sequences, not because there was more of them to start with, but because they amplified more efficiently. And we definitely see that as being a major factor in the very broad groups. So when you're looking at, for example, macroinvertebrates or whatever, you get a lot of this bias. For things like fish, actually, the more we've done it, the more we've done side-by-side analyses. And when I say we, it's not just me and Nature Metrics, but sort of the wider scientific community. The more we see that it's very consistent and it's very replicable. But you have some biases, some consistent biases between different species. They're not huge, but they are consistent. So it scales to some extent with surface area of the animal, just they give off more DNA if they've got a bigger surface area, but it is also driven by things like their activity levels, their sort of metabolisms and things like that. So there's always a bit of variation, and you can in controlled systems control for that and get quite precise actually with estimating the quantitative stuff. When you go out into a wider environment that's interconnected and far more complex and larger, and you've got interactions between species going on, I think that sort of the uncertainty is slightly larger. So we need to always take that into account. What we do see is you can see you can't get to precise numbers of species unless you are looking only at one species. They've done this for salmon in Alaska, where they've managed to calibrate a model so precisely that they can calibrate it to estimate precise numbers of salmon. Generally with metabarcoding, we can't do that, but you do see things like, you've got a barrier in a river and that's affecting sort of upstream and downstream communities. You do see very marked changes in abundance. So you'll get a lot of DNA from something upstream and then much less of it downstream, or vice versa. So there's some uncertainty, but it's not meaningless.
Great, thank you. I would think then, if you focus just on one species, the same species, and you're comparing it in different places, that would take out some of that variation in terms of how the different fish will shed DNA, right?
Yeah, it will. So that is generally easier, but it's also the amount of sequences you get for a species depends also on how many other species are in that sample. So there's a sort of competition effect within a sample which can skew. So you actually have to be quite careful in making those sorts of precise comparisons.
Oh, interesting.
On the other hand, no method is perfect in its ability to give true counts of species. So everything's got limitations and biases, and this is no different.
Great. Yes, thank you.
The thing that I would say on this is I think it's an emerging science in a lot of different ways. And one of the things that we wanted to kind of do is exactly like, so eDNA would work very well if you have a good reference database, very well for presence absence. So qualitative would work really, really well. Quantitative, there are many different factors that might kind of influence the qualitative data, right? The quantitative data. Some species might be, they might be pooling in one area. I mean, how are you going to discern between a live fish presence versus a dead fish presence? All of those different factors are still out there that needs a bit more. We need more studies to figure out the control, cases. And without that, some of these information, they would be and give you a rough estimate of where things are at, but they might not give you the precise information. And so that's why I said it's still a emerging science, especially on the quantitative end of things.
Very good, thank you. Yeah, I think it's I know you all are working on that, and it's still evolving in terms of how to use the monitoring. Great, thank you. There was a question about that kind of links to how far, so in DNA, like in a river, the river's flowing, right? So how would you compare upstream and downstream? Could it be the same fish? How far does DNA flow? How long does it stay in the river? How do you account for that?
I'll jump in on that one. The short answer is that it really depends on the environment that you're working in, and DNA will travel different distances in different sorts of environments. So obviously in rivers you've got directional flow, and so it will move downstream, and the distance that it moves depends how fast the river is flowing. We've actually been, we and several other groups have been starting to sort of map this out in much more detail across landscapes over the last couple of years. So we've got a big project going on in Sweden at the moment, looking at exactly that, at how we can interpret information and how it's moving within a system. And what we're finding is that actually even in relatively fast flowing waters, it's not moving much further than about 1 kilometre downstream in terms of actually picking things up in the community data sets. If you're looking at, if you've got a sort of species-specific qPCR test, which can pick up the tiniest traces of things, then often you'll find, you will be able to detect a species much further down. But it generally gets sort of diluted out and the DNA settles down over a relatively small scale. What it does mean is that you have to be really careful in designing your surveys to make sure that your survey design and the places that you choose to sample are giving you the information that you want about the spatial area that you're interested in. So thinking about whether you sample upstream or downstream of tributaries, or if you've got, I think a particular challenge is if you've got a really sort of point source of pollution or something in a river and you want to know what impact that's having, it can be quite difficult, and you have to have quite an extensive sampling campaign to be able to tease out the impact of that statistically. So we're learning more and more about it, but I mean, that's why we've got a whole team of ecologists and biodiversity scientists who most of their sort of day-to-day work is working with clients to help plan studies and decide where to sample to answer the questions. And sometimes it means that eDNA isn't the right tool for the job. So that's also something that we're quite clear about saying when it's the case.
So one thing that I would add is the utility of eDNA, right? What is the objective of using this technology? What kind of information is it going to get? I think, from a 10-mile high perspective, if we use eDNA, you're going to capture a lot of these different species and all that. And then you can choose indicator species. And I think, and this has been our experience working with the team, like David and Julie and all that. It's actually, other tools might be complementary to eDNA. So eDNA might not provide us with a silver bullet of everything, but it's going to give you an overview of what kind of species there are and what level of abundance and all that. Like in terms of migratory stuff and all that, there are genetics and genomics techniques that we can apply or we can use to figure out the migration and stuff like that. But the larger question would be what is the objective of what we're trying to do? If the objective is to see if certain species, indicator species, are moving up and down, whether they are using the fish ladder or not, if those are the objectives, then we pick up the indicator species or the endangered species in the case of EIA to see if those are there, right? So it really depends the kind of question that you are trying to ask and how well to use eDNA to get the answer for your objective.
Great.
Yeah, you might. It's brilliant actually as a sort of tool for getting that sort of large scale understanding of what's going on, and then that helps you decide where you want to go in and maybe use a whole suite of different methods, or go and actually sort of catch the fish and do more intensive surveys.
Yeah.
Perfect. I agree. Totally agree. I think it's a really great technique for getting that. What is there? Because a lot of our other techniques are not capturing what is there. Our Trishuli data show that we had 7-8 species, and then all of a sudden with the eDNA we have 25. So
exactly.
It gives you a lot to think about, and then you can further look into it. Yeah,
right.
Great.
Right.
Good, thank you. Let me switch to whoops, where's my questions there on kind of the logistics, because that was a good question. Somebody else had how to choose your sampling sites. Were there any guidelines on that? Also, is there any time of year that's best to do eDNA sampling?
I can just quickly say we had a hard time pumping those water when it was a monsoon, because the muddied water and the hard time to kind of push those water through the filter. So I would certainly, and of course in our part of the world, monsoon becomes a big challenge. So you want to avoid monsoon, I guess. But then if you're looking into the migratory stuff and all that kind of stuff, maybe monsoon would be the time also that you want to use. So it really depends. Again, what are you trying to do?
Yeah, and a lot of that comes down to the ecology of the species as well. So again, a lot of it comes back to what's your question. If there are certain species that you need to pick up and they're migratory or only they're part of the year, then you know, target it to that time. DNA does change, so temperature has a bit of an effect on the state of DNA. So it tends to persist for longer in colder water. But it also, in colder water, you often find a lot of species are less active, so they're giving off less DNA. So there's kind of slightly different pressures in different systems, but they're quite, you know, small scale effects.
I had a question about the, did you show that at one point, like when different people were doing the different sampling at the same place, and it showed relatively similar. So do you find like multiple samples from the same site will be fairly consistent, or is there a lot of variation between an actual sample? So how many samples do you really need to take at a site?
Yeah, that's a great question. So again, it slightly depends on your system. So the example that I showed was a, you know, relatively low diversity, quite small river. And in those systems, we basically find everything in every sample. So, you know, one sample, and basically everywhere, one sample will give you a lot of information. It won't necessarily be complete information. So you'll tend to find that the rarer species are more stochastic in how they show up, because there's just less DNA. So it's a sampling issue, right? You've sampled water and there's, you know, you may or may not have caught its DNA. And then when you extract the DNA, there's efficiency loss there. And then when you take a little aliquot of your DNA to put it into the reaction, you know, do you actually pick up the molecules? So the rarer species is the more sampling you need to do to be confident of detecting it. But you know, that completely depends on the environment you're in. So when you go out into, you know, when we've done a lot of work in the really big rivers in the Amazon, you'll get, you know, all of the really common species show up everywhere, and then you'll get a long tail of rare species in each of the different samples. It's the same in the sea.
Same in any sampling technique.
Do as much as you can afford to do.
Yeah, because the best you guys were taking, I think 6 per site, right? In different
Right.
habitats.
Right. And I think, you know, again, this is an emerging science, right? And so initially you take more samples and you exhaust until you can't find anything new. And then you look at the sweet spot in terms of, you know, there is economics that you have to consider as well, you know, because resources are always limited. So you look at the optimum number of samples that you are going to take, let's say in the pool area, in the riffle area, you know, depending on how the hydrology is flowing, how the ecology is looking and all that, the optimized. And this is, again, you know, part of the effort of standardizing your protocol. If you see a pool, you know, if the water is not moving that much, maybe 3 times is okay. But if you have a riffle and water is moving a lot, that means there might be, you know, like less amount of the tissue that are available for DNA extraction. So in that case, you might want to increase your frequency of sampling. So really, again, depends where you are kind of sampling and how the hydrology is looking and how the ecology is.
That's right. There were some questions about like how long does it take to pump in the field, and I think there's a difference between your 2 methods, and also how long can you have the samples before they have to be run? Like how long can the samples last before they need to be taken in the lab?
Kate,
should I start off?
Yeah, you should start off.
Okay, cool. So with our sort of in-field filtering, hand filtering system, once you've sort of done it a couple of times and you're familiar with the process, it takes sort of 15 to 20 minutes to do that whole process. And the amount of water you can filter depends on how much sediment there is in the water. In terms of how long the filters are stable, that depends on certain things like, you know, what sort of preservatives you use. So you need to be careful that whatever you're doing has been tested out with that particular setup. We accidentally stress tested our system in a project in Mozambique recently because we did a bunch of sampling actually with the local community in the coast, on the coast there. And then it took about 3 months to sort out the export permits in that particular case. So the samples were sitting around in tropical temperatures for 3 months. So before they finally sent them back, we sent them out to collect another batch of samples from the same place so that we could sort of see whether there was much difference between the ones that had sat for 3 months and the ones that hadn't. And there was actually no drop off in quality of the samples. So that was really nice. We haven't pushed it further than that at those sorts of temperatures, but it's generally enough time to be able to get them to where you need to.
And so in our case, once we get the filter, we put that in our preservative, and usually we can bring anything from anywhere in the country within 3 to 4 days, a week, let's say, maximum, and they are okay for DNA extraction. But we make sure that when you are extracting DNA, that there is enough of it, that there is a good quality of it, before we kind of do further processing with either PCR or the next-gen sequencing. So there are quality check points on each of these steps to make sure that we're not looking for something that's not there.
Great, thank you. Kat, have you had, what's your experience with export permits getting DNA sent from countries to your lab?
It is getting easier. So now we've got it's only in the last year that we've actually sort of built up this sort of dedicated internal team to deal with all of that. So we've now got pretty good systems and processes. It depends slightly on the country, and it depends whether it's the first time we've exported from that country or not. So generally, once we've done it once, then the next times is very straightforward because we have all the contacts and sort of know how it works. But particularly with exporting, because exporting genetic resources under the Nagoya Protocol can be complicated, and not every country has a sort of clear system and process for that. So our regional coordinators spend quite a lot of time just sort of figuring out those processes to start with. But so far so far we've managed to work out a system pretty much everywhere. We expect there'll be a couple of places in particular that will prove to be more challenging.
Great, thank you. Yeah, it really depends, I think. So there's a set of questions about reference database, the reference collection. So it's great that you're building reference collections in different places, and CMDN building this one for Nepal is going to be very helpful. That's the important thing in order to compare, right? Your eDNA samples, you have to have this good reference collection. So what do you currently use? What is the general reference collection, and how accurate do you think it is in terms of for the Himalayas? Like use GenBank or?
Yeah, so our effort has been that beginning from Nepal Fish Biodiversity Project onwards, we've gone out, we collected, we caught fish, and various different methods we used. And then there is a layer of field-based, identification, fish species identification using some of the keys that we have. And then you pull those together, and then you take the, representative few of these voucher specimens into the lab, and then you do DNA barcoding from their tissue, and so your quality of the DNA, as well as now you have a voucher specimen and DNA barcoding associated with that in place, and then all these voucher specimens are put in a preservative, and we have a nice display of it and all that. Now the key here again is, again, I'm not a fish expert, so we need a lot of help from fish experts, taxonomists. So one of the ways we kind of did that in our first phase was we created crowdsourcing of experts. So we took the video with the pictures or whatever, looking at different, keys, and then we sent it to the experts and gotten their opinion in terms of what do they think it is. And so we would, consult with 3 different experts using crowdsourcing. So we put a database, online database, where we put all of this and requested them to kind of help us out. Now, also we have physics experts from one of our labs, government labs. So we use her help. She's really, really good with identifying fish. So we got a lot of her help as well. And then we also got help from other fish experts as well. So that's important. The morphological identification, the taxonomic identification based on morphology is important, but associated DNA barcoding and identification is important as well, and see how that compares to the global database, either like NCBI, which we use a lot, and then there are fish databases as well, global fish database. So we look at that and see, what are the accuracy in there, both on probability of identification as well as its specificity. Now, if that's not there, then we actually put that into the database. So I think over the years, we've put over 60, 70 different species into the NCBI database. So anyone can access it and get it from there. Meanwhile, we have our own databases, national databases as well, if you would like, right? Where we have the pictures, we have the DNA, and then now when you're doing the eDNA part, then you will be able to look at the global database. If not there, then local database, and see if there is a correlation or linking that you can do for the actual fish that we caught.
That's great. Looks like we had a really good start for the region, because if you don't have all that, it's very hard to use this technique and stuff. That's really great. Let's see, Kat, what's the effort to take to really make a very, what would be needed to, continue with this, what Debesh has started, and really get what the level like you're going for, like you have in the UK, for example, with the whole complete reference?
Well, such a cop-out, isn't it? The answer to every question is it depends. It depends where you are. It depends on the how high diversity the system is, or the country, and what sort of resources you have. I mean, there are some places where there's been a lot of collection done over the years. Unfortunately, a lot of the collections are in formalin, which are quite difficult to get DNA from. And then there's been unfortunately, there's been a huge amount of effort over the last years to do DNA barcoding campaigns. And there's been quite a lot of sort of national efforts to do that, and a lot of money and resource spent. But they've all been driven by the sort of Barcode of Life initiative, which just focuses on the CO1 gene for animals. And unfortunately, the CO1 gene is really difficult to work with from environmental DNA, because you tend to get, if you target, it's very difficult to get precise enough assays that don't amplify all the sort of microorganisms and things. So it means that there's been a lot of effort done that's not that useful. But in some cases, it may well be that the museums or the places that have done that barcoding have got stored DNA that's been extracted from species. And in those cases, it may be possible to sort of cut out a lot of the field effort or avoid duplicating that. So I think that's really worth looking into and trying to follow up. Otherwise, so we've got a big project in Mozambique that we're going to be starting, where we're looking to sort of add 1,000 species to the reference databases there, through the same sort of crowdsourcing method that Debesh was talking about. So we've got a really simple swabbing kit for people to be able to collect the DNA, and then relies on morphological examination, photos and things to try and put a taxonomic ID to things.
Great, thank you. I think people probably are thinking, how much does all this cost to do these kind of projects? Debesh, do you have a ballpark of how much it costs to do like maybe the February sampling that you just did, plus the running the samples kind of thing? And Kat, how much are your kits cost? Or
so maybe I can give an example of the Trishuli project that we did. think it was like 7 different sites. And I believe there were 2 different seasons, and the overall project cost was, I think it was like a little over $90,000.
Great, thank you.
So yeah, it depends what you're including in the cost. So our costs don't usually involve the sort of field mobilization, but for us, we charge on a per sample basis. So it's £275 per sample, and that's for the kit, the DNA extraction, and then the analysis of one group. So that might be fish, or it might be vertebrates, or it might be insects, or whatever. And then there's, if you want to have a second analysis run on the same DNA, obviously we don't need the costs of the kit and the DNA extraction and things again. So then it's sort of an extra about £100 per additional group.
Great. And how there was a couple questions about using this for like insects and vertebrates and other vertebrates. And have you, and I think you showed it to be fairly useful for all these other things. Do you see any particular limitation in using it for these other groups?
Yeah, I mean, you have to be much more careful about how you interpret things quantitatively from the other groups, because they're putting their DNA into the system in different ways. You know, for example, in the Amazon, we see species like tapirs are massively overrepresented because they defecate in the rivers. So the river is sort of full of tapir DNA, whereas other species you know, have different habits, you get much less detection of. So you have to interpret those much more as sort of incidental detections and sort of understand the ecology as much as you can. I think freshwater invertebrates is an area that there's been a lot of work on and has been really challenging to get macroinvertebrate eDNA up and running, partly because just when you know, making that assay, which is broad enough to get all of those different taxonomic groups that make up that group, but without getting the things you don't want, like bacteria and copepods and things that are at much higher concentrations, is really hard. But we've had a bit of a breakthrough in the last year, and actually finally are able to get really very good freshwater insect data from eDNA. So that's it's still evolving a little, but we've gone from saying no, it's really not possible, to saying actually it's looking like we can start to get some good data.
Can't hear me. Okay, great, thank you. So this has been really great. I mean, it's so exciting, this whole field and the uses. I mean, I know there's a lot of things still being worked out, but it's such an exciting field for using this unique technology of using the genetics to detect species. It can cut down on sampling time in the field. It can get us a lot more information, even just to start us off, you know, to get that presence data, absence data, to then be able to have an idea of what's there. And then I think the techniques for looking at this quantifying and comparing over time are really going to just keep improving. So thank you both very much for sharing with us today. It's been really great. And I hope to see a collaboration between you two and all of us together to keep doing this in Nepal and building the database. And I think there's great use for it, particularly since we really need some monitoring around all the hydropowers going on in Nepal. So great, thank you so much for taking the time to share with us today. It was really interesting. And I just want to remind everybody of what's coming up next. There are 3 more webinars for the workshop on the Trishuli assessment tool, macroinvertebrates on Thursday, and then Himalayan fish identification, which will be really interesting next Wednesday. It's on Wednesday instead of Tuesday because I think there's a holiday in Nepal on Tuesday. But it's going to be very interesting to look at, you know, how are we linking, what are the status of taxonomy for all these fish that we're then identifying with other methodologies. And then I'm going to attempt to talk about data analysis on Thursday. So hope you'll join me then on the 18th. And then in March, we have some really interesting webinars coming up by David Phillip and Julie Clausen on radio telemetry and tracking fish movements on March 16, and then about on hatcheries, rethinking hatcheries, review of the costs and benefits. And this is a additional webinar we're adding, a bonus webinar to the series on March 23rd, and we'll send out a flyer. So so I'll just end there and thank everybody for joining us, and thanks particularly to the speakers and everybody who helped out, and I hope to see you on Thursday. Great.
Thank you so much, Leanne.
Thanks everyone.
Take care.
Bye.
Bye.
Bye.